Hi everyone, welcome to today's webinar, Accelerating Biomarker Discovery and Therapeutic Development, Customized Solutions and Drug Repurposing. I'm Iman Bhattacharya, Senior Global Product Marketing Manager at QIAGEN's Bioinformatics Group, QIAGEN Digital Insights. We are very excited to have you join us. If you have any questions during the webinar for our speaker, please use the Q&A panel for the questions. And today's topic is very timely, and it's about speeding up the path from discovery to therapy. We will explore how customized bioinformatics solution can help researchers and translational researchers uncover new biomarkers, accelerate drug development, and even identify opportunities for drug repurposing. Our discussion will touch on large-scale single-cell and spatial transcriptomics, multi-omics data integration, and AI machine learning-driven approaches and tools that are transforming how pharmaceutical and biotech companies find and validate therapeutic targets. By the end of the webinar, you'll have a clearer view of how these strategies can save time and resources at critical points in drug development pipeline, and how they are already reshaping the way we approach precision medicine. Our speaker today is Dr. Sathiya Manivannan. Currently, he's a scientific consulting expert at our group, QIAGEN Digital Insights. Sathiya works closely with pharmaceuticals and biotech partners to deliver tailored solutions that speed up therapeutic target discovery and optimize drug development. His expertise spans large-scale single-cell and spatial transcriptomics, multiomics, database generation, and creation of graph knowledge bases and AI ML-driven analytics, all designed to help companies to save valuable times and resources at critical stages of drug development pipeline. Before joining QIAGEN, Sathiya served as a bioinformatics analyst at Genentech, where he built efficient sequencing data pipelines and designed interactive single-cell analysis tools to support therapeutic program. With that, I would hand it over to Sathiya to take this ahead. Sathiya, welcome. The floor is yours.
Thank you so much, Iman. I am excited to share with you some of the stories that we have in multiple collaborations that we have had with our customers in building tools, knowledge bases, and solutions that are customized for their therapeutic discovery and transferring discovery into translational applications. And today I want to share with you three main topics, and starting with how we could achieve drug repurposing in the field of oncology using our knowledge bases. A legal disclaimer, this presentation and the solutions discussed in this process are only intended for molecular biology applications. They're not intended for diagnosis. And all the product-specific information and disclaimers, please refer to QIAGEN.com and the technical services. So over the last 5-6 years that we have worked with our pharma company, the landscape for data analysis has tremendously changed, both in thanks to AI ML developments, but also due to the volume of data that is being generated. But overall, the pain points in data analysis and therapeutic development has remained consistent and ever expanding. The first and foremost request or conversation that we have with our collaborators in pharma and biotech is how can they maximize the drugs that are already being approved by FDA, that are already in the market, that are safe for patients to take, or is in the final stages of development that pretty sure it's going to be approved. And how can you see if that drug that is highly valuable, a lot of information has gone into, can be repurposed and used in a different scenario, a different disease indication, or could be used in combination with another drug or a panel of drugs. The second most common conversations we have with our customers involves developing new pipelines. As data becomes more complex, as multiomics expands from genomics, transcriptomics, proteomics, and metabolomics, we start looking into issues of integrating these data sets into a seamless and uniformly processed data set and databases that can be then mined both by human and by artificial intelligence tools. To do this, we need not only the data itself processed uniformly, that also scales with the size of the data, but also we need the metadata to be very carefully curated. And it requires a team of data collectors, pipeline builders, and testers that can all work together as a unit to develop these pipelines. Also, once you have the data, it is also important to understand how your data compares to other data sets, not only within your team, but also in the public space. This can add tremendous value to your data, but also can weed out issues that you see with specific batches of data. You can also expand your analyses by incorporating these publicly available data sets, but also doing a very clever way to search them and mine them for specific information that you seek. I don't have to go through the stages of drug development for this crowd. We all know that the drug development continuum lasts anywhere from 10 to 15 years, and it can start from a simple idea that a specific drug could be used for a specific disease, or could be starting from a molecular discovery of a specific target. In either case, the lead identification, lead optimization, candidate selection, all of these steps constitute the discovery phase, which is often the highest or most expensive part of the drug development process in terms of time. It also has the highest risk stage, where you can create a lot of candidates that do not survive past the candidate selection phase or do not end up being a good candidate. But failing to do this step carefully can be very expensive, because the failure rate downstream is low, but the cost that has gone into failure at that stage is pretty high. So for example, if your drug is really good in vitro, but fails in safety, you have already lost quite a bit of time and resources that you have spent on it. If it is safe, but its efficacy is only moderate, you have gone to clinical phase 2 and have again lost millions of dollars. So one of the things that people have been asking us is, we already have drugs, multiple of them, that have been approved by FDA, that has been clinically evaluated for their potency. What is the drug concentration we have to use? All of this information is available, but can we apply it to a different disease or a different context along with another drug? And this can be achieved. This is being done in multiple cases, either through the iterative process of experimenting in new cell lines or new disease conditions, or more and more using knowledge graphs. What is a knowledge graph? Knowledge graph is a densely connected network of information about genes, drugs, diseases, and their functions and pathway, all interconnected through carefully annotated connections. This is a way of looking at the holistic information of all the knowledge that has been gathered over a long period of time in the biomedical field by a number of experts in each of their domain. And these knowledge graphs can be used not only to understand the pathway better, the disease's role better, but also can be used to predict connections that have not been there before. And this drug-gene association and drug-disease association can be highly valuable in creating hypotheses. And we have had several customers working with us and creating these kind of connections, and being, more importantly, able to validate those using small experiments. What do we have from our product side is QIAGEN Biomedical Knowledge Base, or BKB. This is a carefully curated network of genes, diseases, targets, and drugs that has over 24 million relationships which are being carefully collected and curated by experienced PhDs and MDs in those respective fields, and they have been made available in different formats. We also have an expanding network of relationships, over 643 million, created using the BKB AI, which is a LLM-supported curation and collection of these information. So what can you do with this knowledge graph? You can start by identifying what is your gene of interest or your molecule of interest and create a subset of those networks. And then you can start doing hypothesis hopping, just like finding what is the disease, what is the current drug, can I use this drug for something else? Can I augment the existing drug with an additional drug that complements the mechanism? This can be highly useful if you're thinking about treatments where the resistance for a specific drug develops really fast. If you can also simultaneously block the resistance mechanism using a different drug that doesn't conflict with your drug, that can be of great help. You can also work on identifying the side effects of using this drug and trying to mitigate that by using a complementary drug. This can also be achieved using several relationship algorithms, also by re-embedding some of our knowledge graphs into different models available out there. The second topic that I want to discuss with you is pipeline development. Pipeline development requires very carefully curated set of tools that can not only work at the level of the computer infrastructure, but also make sense in transferring information from one step to another. So what do I mean by that? Most of our pipelines, whether it be simple RNA-seq pipeline or sophisticated neoantigen discovery pipelines, all rely on the same principles. It requires you start with very carefully curated data collection. The data has to be supported by carefully curated metadata information so that the data can be sanitized, remove any kind of noise from it that is technical, but also understand what the pipeline needs to do with these complex data sets. The second step is selecting tools. There's a lot of open source tools available. We have also built some tools from scratch using algorithms that we have developed with our customers. But these tools not only need to be containerized, meaning they work the same across different platforms, but also they have to be benchmarked against each other to make sure that the tool works best for what you need. Next, we create unit tests, making sure that these individual containers and the tools work as they should. And this can be an iterative process requiring a lot of manual effort, ranging from 1 to 2 months of time spent on each tool. The next bigger challenge is chaining the tools together and optimization. Each tool has its own input and output requirement, and we often end up trying to figure out what is the best way to convert them without losing noise from one tool to another. And then we perform integrated testing, and the output is manually evaluated by a team of experts in that particular domain. Not only this, but eventually after delivering the pipeline, we often train our customers to know how to run these pipelines on their end, making sure that they are also able to catch fringe data set issues. The errors that are there finding from their end are also properly supported, and this requires continuous pipeline maintenance. This typically requires 1 to 3 member team and can require a lot of manual effort to develop. What we bring from professional services is an advanced team of not only biologists, but also computational biologists, engineers, and bioinformaticians, all working together as a team. And whenever there's a customer project that involves pipelines, we bring in the experts of the individual domain and make sure that we are looking at the pipeline development, not just as a series of code, but as a resource that will be used for future purposes and will be applicable and usable for the customer for several years together. One such example is of us using an existing open source pipeline from NFCo, Spatial VI. This was one of our customers' requests to modify this pipeline so that the data that is coming out of this open source pipeline can be very carefully integrated into their data set. We scoped this project and we were able to deploy this pipeline within 2 months of time and then test it in their environment and create data sets and reports that were highly useful for them within 4 months. What did we do? We started with creating spatial data sets from public domain. The customer was unsure if they could use this data set for their research purposes. So they wanted to try publicly available data sets. We collected the publicly available data sets, added metadata to it, processed it using the public domain tool, but also added data set formats that could then annotate this information, the information coming out in higher depth, in higher quality. So, for example, identifying regions of higher malignancy score, identifying interfaces of stroma and cancer, and identifying each cell type within those zones, and trying to find relationship between these interacting cells. Overall, the report was not only customized to what the customer wanted, we were able to refine the pipeline parameters and expose the relevant ones to the customers and not have all the ones that can be abstracted away from the customer. We also added new tools for visualization and exploration and made sure that this was compatible with other tools that they were using internally. Overall, our team was able to provide end-to-end support with not only out-of-box solutions, but also customized solutions that help them improve their pipeline efficiency. One of the tools that often we find our customers using for data exploration in the single cell field is BioTuring. While BioTuring has extensive support on their end, customers often find that their pipelines that they want to build and maintain are not necessarily directly compatible with BioTuring or cannot be deployed in BioTuring environment and run within that environment. They also find that public data collection and curation often is different between different vendors, and they often ask us to find out whether there is a way for us to convert one data format to another one. And this is where we excel because our metadata curation process, our data pipelines are usually versatile and can be plugged and played in different environments. We also help you import data sets into these kind of other vendor provided tools that makes it possible for you to work with tools that are built for QIAGEN and QIAGEN solutions with external vendor tools without a lot of interconversion. And downstream of whatever you develop from BioTuring or other pipelines that you use for analyses can also be then used for systems biology analysis using our products such as IPA ingenuity pathway analysis. The third topic that I want to discuss with you is about how you can utilize highly harmonized, publicly available data sets that have been uniformly processed and have been curated to the same standards, and use this not only to explore the publicly available data, but how to use your data set along with them to create highly informative inferences. One such analysis that is possible, and professional services has done projects on this topic, is differential expression matching to identify common effectors. Often we find pathways and differentially expressed genes vary between different data sets because of batch effects. Often these batch effects are attributed to difference in the original sample, or often to that of the method that was used for sequencing or the depth of sequencing. These batch effects are often corrected and can be fixed here and there, but sometimes there is some information in the batch effect that might be useful. The batch effect can often indicate underlying variations at the level of genetic information that can cause the differences. So one such project came to us from a customer who wanted to know how their samples, their cell lines that they are using for their xenograft models differ from that of publicly available in vivo samples. Are there xenograft specific mutations or causal drivers that can affect the differential expression that they are seeing that is not observed in publicly available data sets? To process this, we collected their data set, processed it through our pipeline, identified the differential expression analyses, but also used the RNA-seq data to identify variants, SNPs, and indels. And then we were able to compare these SNPs and indels to that of the publicly available data set and identify variants that are common, that are unique to their data sets, and then use system biology approach to identify whether some of these variants that are unique to their xenograft model can cause some of the differential expression that they're seeing or impact the differential expression pattern that they're seeing while that is not observed in public domain. So these kind of comparative analyses can highly enhance what kind of information you can get from your pipelines. Using such approach can also help you identify variants that can reveal neoantigens. Neoantigens are popular candidates for drug development because they are often immunogenic, so they lie at the interface of oncology and immunology. But creating these kind of therapeutic drugs cannot be done without a large collection of data set for each of the diseases that you want to analyze. They have to be also processed in the same way. You have to identify variants and have a pipeline that filters out common variants. And you have to have a pipeline that evaluates whether the variant does lead to a protein. Because if it is a variant that affects expression and doesn't make a protein, it's not immunogenic. And finally, you have to have tools that can calculate the immunogenicity of that particular protein and whether that protein is available for you to access through your immune cells or immune biology tools. Standard pipelines can take all the way up to 2 years to just develop and implement these solutions. But with QIAGEN's out-of-box solutions that can be chained together in different ways, we have achieved something like a neoantigen discovery pipeline in 4 to 8 months for our customers. In summary, QIAGEN Digital Insights products plus professional services can offer novel solutions that can help you tackle not only current challenges, but also future challenges that you encounter. This includes indication expansion through the use of our knowledge base to create novel indications for drugs or expand drug combinations that can be used against existing resistant development in oncology. We can also use our tools to help discover neoantigens using fusion detection. Also identify commonalities between your sample and something that is available in public. And in addition, we can also create knowledge bases from your private data embedded in a data set subset from our knowledge base, creating a hybrid system that works efficiently and also can help you fine-tune genomics foundation models that you might want to use for your AI/ML application. With that, I would like to thank you for participating in this webinar, and back to Iman.
Thank you, Sathiya. That's a great presentation. And I personally was so delighted to specifically see where you're talking about accelerated reliable neoantigen discovery using the comprehensive and growing omics knowledge and databases. So it was really great just to listening to you. We have few questions coming in, so we'll open up for the audience questions now. And I have a few great questions. I'll just directly read it to you, and you can take your time to answer them, okay?
Okay.
Sounds great. I think the first question is, how does drug repurposing using knowledge graphs compare in efficiency and accuracy to traditional methods of therapeutic discovery?
It's a great question. So one of the things that often is a bottleneck for traditional drug discovery is trying to find the right kind of setting or the right kind of system to try the new drug development. For example, if you have found that your drug works really well for a specific type of cancer, now you want to see if it works for a different type of cancer. You have to now cultivate that specific cancer's in vitro system, create a new cell line or acquire a new cell line, collect data from it, make sure that you understand the basic biology of that particular cell line, and then try the new drug. Sometimes people do so by observation in clinical reports where patients with multiple indications have been identified to respond positively to a drug, and these are serendipitous. What we are doing with the knowledge graph is that we are providing the context where these drug repurposing can be efficient. It doesn't remove the necessity for validation in different systems, but it can narrow down the scope and also provide a mechanistic backing for it. When you try the drug on a new system, it may work the same way or may not work the same way. But with the mechanistic background, you now have an idea of evaluating whether the specific targets are being affected, how they are expected to be affected, what are the side effects that we can expect, and whether those can be evaluated at the level of expression or assay development. So in summary, what our knowledge-based driven drug repurposing helps you do is to reduce the number of iterative tests that you have to do before a positive indication can be achieved. And when you do have a positive indication, what are the markers you need to look for evaluating that drug in the new context?
Thank you. That's a lot of validation to pharmaceuticals and biotech researchers. Thank you. I guess my next question is, how do you balance the need for customization in analysis pipelines with the scalability required for large-scale studies?
Yeah, this is a great, fantastic question. Often we find our customers coming to us with pipelines that are legacy pipelines developed 5-6 years ago that they want to apply on their new data set, and it doesn't scale well. It needs to be modified. The tools are not properly established in there. What we do is that we first look at the pipeline and find out the steps that can be modularized. What do I mean by that? If there is a QC processing step, that doesn't have to be integrated very tightly with the alignment step. You can separate them into 2 separate modules, build pipelines that work independently for each of them, and then integrate them. Once you do that, the scalability question is kind of abstracted into it, because for each of those tools, you identify the resources required and find out the best way to run them in parallel. When you build tools together, we have also looked into publicly available open source tools like Nextflow, SnakeMake, and we have adopted them and made sure that those can be run from a graphical user interface so that the burden of developing something in command line goes away from the customer. And finally, we not only deliver the pipelines, but also support continuously the implementation and execution of the pipeline. This often means that we identify the bottlenecks that the customers are facing and find solutions not only to improve the infrastructure that the pipeline is running, but also identify code enhancements that can improve efficiency.
That's a great answer. Thank you again. And I think in the interest of time, I will take one more question. This is specifically on the customizations on the knowledge graph part. So the question is, can users or the researchers customize the knowledge graph to prioritize specific disease pathway or molecular targets?
Yes, precisely. So often we get requests where they are specifically interested in one drug target or one specific molecule that they want to further study. And what we usually do is we subset that graph and create different kinds of embeddings with different modularity. What do I mean by that? When a specific drug is looked in the context of how it relates to the disease versus a specific drug, how it interacts with different genes, can be layered as two separate layers within the knowledge graph. So once you have the gene-gene interaction as a separate layer, you can now start adding information to that layer without affecting what you have already in the gene-disease layer. Now you can add a third layer of disease-disease interactions or disease mechanism commonalities and start building the knowledge graph in layers that helps you highly customize it for your specific disease, target, gene interaction analyses. And there are multiple tools that can be used to not only predict missing links or predict drug-drug interactions that can reduce the efficacy of your drug, or even go into the level of predicting what would be the perturbation effect at the level of gene expression. And these simulations, these predictions can provide a lot of insight into how the drug is impacting or interacting with a specific disease.
Thank you. That's a great answer. So with the interest of time, we will not take any more questions. Thank you, Sathiya, for the great talk. And if you have any more questions which we didn't get to answer, if you have any other general questions about our solutions or our service or the products, please reach out to us. Sathiya, would you mind go to your last slide, to the contact information provided over here, or go to our webpage, www.digitalinsights.qiagen.com. Sathiya, once again, thank you so much for your talk today. And with that, we'll close out and have a great day, everyone. Thank you.
Thank you, Ima.